Hello, everyone. We're Hans and Perry, and today we're going to introduce the topic of bacterial growth. So, bacteria, a single-cell microorganism, can be found in every corner of the globe. In fact, it can be performed in various shapes, and it can be divided into two different types, which are pathogenic and non-pathogenic bacteria. And today we're going to see three pathogenic bacteria that can spread disease. So personal interest. So school is an ideal place for bacteria to start as children create a constant supply of microbes. And according to the data, more than 2.7 million of bacteria per square inches are found in school surfaces. So hence, as a KCIS student, we aim to create a germ-free environment through investigating under what conditions does the bacteria can thrive. In global context, one of the most common pathogenic bacteria is E. coli bacteria, in which the water availability is critical for it to grow. In class, we learn about diffusion, which is the movement of molecule from an area from higher to lower concentration. So, in fact, diffusion is essential as it can help water, oxygen, and nutrient to transfer to bacteria for bacterial metabolism. So, our research question is how does different environment, bacteria plate, moisture level, 0, 1, 1.4, and 2 ml of water will affect the bacterial growth, because we will measure the bacterial growth through investigating the total grid paper units the bacteria occupied. And hypothesis. Our group hypothesis that the increase of water level will accelerate the bacterial growth, in which the order will be 0 to 1 to 1.5 and 2 ml. And this is because high moisture encourages tomato opening, in which allow bacteria to improve cuticle permeability. And so the variable. So our individual variable will be different amount of environmental moisture level, including 0, 1, 1.5, and 2 ml, controlling by adding different amount of water with a dropper. And dependent variable will be amount of bacterial growth, in which we will investigate through counting the total box unit of grid paper and then calculate the percentage of the surface area. And so the control variables. So we'll be using the same type of nutrient agar and same type of cotton swab, and also constant location, including same rule to grow bacteria and same bacteria collecting location. And we'll also have constant amount of nutrient agar, in which each petra place will have 12 ml of nutrient agar. And the procedure. So we'll be collecting the bacteria with 12 individual cotton swabs at a school bathroom toilet, and we'll put 12 ml of nutrient agar into 12 individual petra dishes, and then label the moisture level on 0, 0, 1, 1, 1.5, and 2, and we will swipe the cotton swab on each nutrient agar plate, and then we'll place the grid paper below and observe bacterial growth for 12 days. So as you can see, we will be conducting 3 trials, and then we'll be adding water supply once each 2 days, based on different moisture level we set. And the safety precautions. So due to ethical reason, don't let others to directly attach a cotton swab with pathogenic bacteria, which can potentially make them be infected. And then also due to environmental issue, we should dispose correctly using a plastic bag, so don't harm your community. So, and due to physical reason, you should wear gloves when attaching and collecting the bacteria on the toilet.
So moving on to the pictures and the results. So looking at the picture on the left, all bacteria have no signs of bacteria growing. On the bottom right picture, signs of bacteria growing has been shown. So data table and graph. So the data of the left records bacteria growth under different environmental moisture level as time passes. The figure on the right is the graph, which the horizontal axis is the number of days passed, and the vertical axis is the percentage of space occupied by the percentage of space of petri dish occupied by the bacteria. So conclusion. So looking at the graph, no signs of bacterial growth is shown between day 0 to day 4. From day 6 to day 14, bacterial growth grew in both 1 ml and 1.5 ml water, reaching a percentage of 45%, which bacteria growing in 0 ml water only reached 25. Taking a further look at the graph, so growth rate of both bacteria was 1 ml and 1.5 ml slowed down. So hypothesis evaluation, what we hypothesize is halfly correct, because from the 1 and 1.5 ml water has decent amount of bacterial growth. However, bacteria growing in 2 ml water has no signs of growing. So, first, we have 3 trials. 3 trials enables us to find out the outlier and come up with the most accurate data. And the small scale for dropper. So this enables us to obtain small amount of water. Usage of same nutrient agar. This enables us to give the bacteria the same type of nutrients. Weakness. So did not mention how to prevent the petri dish from being contaminated from other bacteria. This might lead to inaccurate bacteria growing. Also did not mention how to precisely measure the amount of bacteria growth. This might lead to inaccuracies as miscalculations may be made. Limitations did not equally distribute the amount of nutrient agar on the petri dishes. And did not strike the same surface area of the toilets. Did not strike the toilet surface area with a cotton swab with a certain amount of times. Improvements. Make sure every part of the petri dish are covered by nutrient agar so all bacteria have the same area for growing. Set a certain area and only swipe the certain cotton swabs over that area. Set a certain time for swiping the toilet. For example, swipe 3 times with the cotton swab. Extension. Besides environmental moisture level, other factors like the environmental temperature as well as nutrient concentration can also affect bacterial growth, as shown in the 2 graphs. Therefore, we want to investigate how these factors also affect bacterial growth. Last, application to be rolled. So, as shown in the experiment data, humid environment can lead to rapid bacterial growth. Therefore, it is necessary, especially in bathroom. Therefore, it is necessary to keep our bathroom dry. Having an adequate ventilation, installing fan and using dehumidifier are all methods to keep our bathroom dry. This is the end of our science fair Pechakuchen. Thank you for listening.